Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Our first talk will be given by Marty Humphrey at the University of Virginia. It'll be Model-Driven Cloud Services for Cancer Research. One reminder, during the Q&A, if you can wait for the microphone, we're using it to actually record the questions because we're videotaping in the back. That would be great.
Okay, thank you for coming. This is going to be somewhat of a nuts and bolts talk. I'm going to show Visual Studio. I'm going to show other development tools. You won't really see so much code, but I wanted to preface it that way. The context, what we're doing here is in collaboration with the University of Colorado Health Cancer Center. We participate in this activity called CA Big, which is the National Cancer Institute's bioinformatics grid. What they're trying to do is share data across the centers, the cancer centers, more easily. Okay, so there are a number of participants, and this is the web portal for the whole thing. Now, so what you need to know about CA Grid for this particular talk is that it's unique. From my perspective, it's unique in that it's very much driven by semantics. And what I mean by that is not only is it syntactic requirements with regard to the service-oriented architecture that they're constructing to try to share information. That's the syntax part. The semantics part is that they have a controlled vocabulary across the entire lexicon regarding the cancer research. And so, for example, if I'm going to try to make a certain set of data available, and part of it has something about a gene, then I have to represent it in a canonical way. I can't sort of stand up my own idiosyncratic definition of gene. They're trying to do this in the wide area. They're trying to support site autonomy, that kind of stuff. And so it's very challenging, somewhat unique from my perspective, that they're really tackling semantics. I mentioned it's a service-oriented architecture. What I'm going to focus on today is what they call a data service. The nature of a data service is what you might imagine. It's trying to provide a collection of data to the cancer research community. And it's important that it is a web service. It has a standard interface. It has a particular single interface. Okay, it accepts a particular query language called the CA Grid Query Language, CQL. Okay, and so there are all these services. They have the same web services interface, but they actually, of course, they have different data behind them. Okay, and so that gives you a little more context of the setting. So more particularly, what we're going to do here is this is the particular challenge we're going to face. This is the challenge that colleagues at the University of Colorado are facing, and we're trying to help them out. They have a particular collection of tissue samples. They have this encoded in a SQL Server database within their enterprise. They now want to make these samples available to CA Grid. Okay? And so one of the problems that the fundamental problem they have is, how do I do this? How do they put this out there, conforming to, in effect, the effort of sharing tissue samples that they call the common biorepository model. So what they need to do is be able to still have their database, but it needs to conform to this biorepository model. And we're going to show some stuff that we've put together to try to do this. An overview of architecturally what we've done to try to tackle this is this is what the service is going to look like. And what we're going to try to do is create the service that will have their tissue samples in the back end. Outward facing, it will have the CA Grid service interface, and we'll have the implementation here. I mentioned this thing called the CQL, the CA Grid query language. So we're going to parse it here and serialize and deserialize. We're going to do a CQL to SQL mapping to get into the data. So the client's going to come in, hit this canonical interface, We're going to do some parsing here. We're going to tap into the database, and we're going to ship it all out that way. Okay? Now, so you'll see that this is actually a generic data service for CA Grid. Now, so if we want to conform to a particular model, in this case, the common biorepository model, okay, there's a certain amount of this that's generic, and then there's a certain amount of this which is specifically tailored to the common biorepository model. So our challenge that I'm going to step you through is how do we do this as essentially as painlessly as possible. And the way we do that is we take our model of the common biorepository model. We're going to generate specific code that we're going to layer in here based on that particular model. The two files are really a collection of functionality regarding the biorepository model, the CBM. And then we have some mapping that's just declaration regarding metadata on that service itself. This one is not particularly interesting. We're going to focus on that for this talk. Once we generate the code that is specific to this, then our second half of what we're going to try to do is actually deploy it. And so our second half is focused on Azure. And so what we're going to do is take our DLL and our config file, upload it into blob storage on Azure. Then we're going to configure the generic grid service for Azure to read the customizations from blob storage. And when it boots up, it'll magically work. Now, so Wright, so the whole thing is we're going to do it from scratch, too. And so we'll see how this goes. Don't worry, I'll talk at some point. Now, so I've constructed a virtual machine with all my goodies on it in Amazon. Why have I done that? Well, Azure doesn't give me this infrastructure. This infrastructure as a service yet, so I use Amazon. So first thing I want to do is show you a typical client. In other words, how we might actually search for common biorepository model, or in other words, looking for tissue samples. So we wrote an application. Let me find it. So this is our typical application that a cancer researcher might use. They fire this up on their desktop, and they're going to probe the services that they know about looking for particular samples. So I want to show you how this is done. This is before we stand up our service, of course. So we have configured it to know a couple services. This is one that's running at Ohio State. We give it the service endpoint. We then ask the service for the model that it is serving up. So this is actually going to go out to Ohio State, ask for the model. When it gets back, we're going to construct a query interface based on that model. And so we're going to, let's see if I can do this. Oh, right. Hold on a second. I'm going to install ZoomIt so I can come in. So what we're going to focus on is the specimen collection summary. Let me get out of this. And we're going to ask our favorite query, which is to look for samples related to the prostate gland. And so this is our base, and we're going to add a criteria to look for a particular sample. I pulled down the wrong one. Participant collection summary. This is the problem with live demos. Specimen collection summary. Specimen collection. Well, we'll just ask this sample. This is we'll ask for the participant collection summary of this service, and we won't provide any other constraints on it. And so we'll submit the query to the service, and we'll get these collection of samples back. Okay. So this is in some sense, this is our legacy client. And so the question is now, how easily can we stand up a service such that we can now aim this legacy client at it? And so let's step through the necessary steps to do that. So the first thing we need to do is we have our database on this virtual machine, and what we're trying to do is make this available according to the model of the CBM, the Common Biorepository model. So let's step through how to do that. We're going to fire up Visual Studio. And what we're going to do here is use some new support in Visual Studio for model-based development. And we're also going to combine that with some code that we've written at the University of Virginia. So what we want to do is create a new modeling project in Visual Studio. And we don't really care what the name of it is. The particular modeling support that we're tapping into is UML support. There's other support in the entity framework. We're not touching that at all. So what we want to do is we have previously downloaded the Common Biorepository model, the UML for the model. The XML representation is actually XMI, so we're going to load that in. And so we've loaded in the model, and I'll show you a little bit of what it looks like. I'm just expanding the model, and what I want to look at is a particular two classes, and the way we can do this is by creating a new item in the project for UML class diagram. And now we can visualize a couple of the classes. We'll look at the participant collection summary. And we'll also look at collection protocol. Okay, so this is, again, what we've shown to this point is new support in Visual Studio. So we haven't really done anything yet. And what you can do is you can start exploring your model based on the UML support that's just coming in Visual Studio. Now, so again, our problem statement is we have this model, we have back-end data. We don't know if they are consistent. And so what we've done at the University of Virginia is we've added support that checks whether or not a particular back-end data source is consistent with the model. And so in a moment, you will see a dialog pop up that allows us to specify which particular database that we want to try to expose via this model. And it'll be popping up in a second. There we go. And so the server, I mentioned that the database that we're going to look at is on this machine. And so the server is localhost SQL Express. And we'll type in my username. We'll hit the connect to see what databases are actually there. When it comes back, this will be populated. The actual database that we're looking at is one called tissue samples. When we hit the done, our code that we've written is going to check that database against this model. And what we come up with is shown at the bottom here. Let me scroll up. And it says the error, a particular table does not have a particular column, and we have another error that has a particular table that has a problem with the column. Now, clearly, a disclaimer: we've tried to make it that the database is pretty consistent, and if you really start with a database that is wildly inconsistent from the model, then we can't help you right now. But we have code, and we're expanding it to try to make the matching between the model and the database much more sophisticated. But let's just look at these two for a second. It says that our particular there are two problems between our model and our database, and so what we're going to do to make them consistent is we're going to change the two problem classes. One of the classes is right here. What our checking is saying is that one doesn't sync up with what the database expects. So we're going to change that directly in the class diagram. And our second problem, this was the participant collection summary. Our second one is a specimen collection summary. And so to change that, we're going to drag this out onto the pane as well. Scroll over. This is the other problem. And so we're going to now change this to be consistent. We're going to rerun our check. And we'll see that we have no output indicating that our model is now consistent with our backend database. Okay? Now, so next thing that we've done is written code. In some sense, this is where the magic comes in. We've run code to generate a service, essentially based on the model. And so the code down here, which I won't get into, is essentially the code necessary to have the external interface that the CA grid expects map within the service to the common bio repository and to tap into the database in the back end. That's all the code that's necessary. So what we can now do is we can build this project, the library that we just created. And we'll see that there are no failures here. So at this point, we have created the necessary functionality based on the model. And so our last step is to deploy the model, or deploy the service that will serve up the CBM data. I mentioned that we were going to do that via Azure. So to save a little time, we'll also do it via the development fabric. So we want to start up the development storage on this desktop. Then as that's starting up, we want to use a tool called the Azure Storage Explorer, which is in CodePlex. This allows us to manipulate blob storage in the back end. Takes a moment to boot. Takes a moment to boot. There we go. So again, this is a tool that you can use against the development fabric, or you can use against Azure itself. We're going to use it on the development fabric. What we need to do is create a new blob container. We're going to call it CBM. We're going to create that. If you recall, I mentioned that we had to upload two pieces of information. That is the customization of our generic grid service. And so we're going to do that now. The first one is the DLL that we just created. Documents. Well, actually, the first one will be the configuration file because I came across it in the dialog. The second one will be the DLL. Upload. And so this is our Visual Studio that we just made. I'm looking for the DLL that we just created. And there it is. So we've uploaded our functionality. So the very last step is that we now actually have to deploy the service. And so let's do that. So we just have to deploy the service, and we have to configure it to read from the blob storage that we just uploaded the custom DLL for. Okay? Strictly speaking, we don't have to use Visual Studio because we're just deploying it. Visual Studio provides a nice way to customize the service we're about to deploy, and so that's why we fire up Visual Studio. The way we configure it is we pull up on one of the roles, the Azure role, we pull up the properties. Let me get rid of this. Come on, come on. Why the hourglass? Why the hourglass? There we go. Right. So these are the settings that we need to change in order to configure the service. This is a generic service. We're going to make the modifications to use the CBM DLL that includes the connection to our particular backend. We need to make a couple changes. First one is what is the name of the blob container? That is CBM. Notice that there's a couple configurations here to use development storage, indicating that we're using the development fabric. We're not really using the actual Azure itself. We need to configure it to read the particular DLL, which is modeling project 1. The last one we don't need to change. And then the last step we need to change is the actual connection string to the backend database. And this is where I've been getting it wrong, so we'll see if I get it right. Okay, so we've configured it. Now we're going to deploy it. F5 deploys it. F5. I must have cleared the output screen. It's packaging the service deployment down here. Attaching the debugger. Again, we're going to Azure Development, which is local machine. It's going to attach the debugger, in this case IE. Well, now, if we get a nice web service screen here, well, that's a good start. So that means that the service is up and running. It doesn't mean that we've made the proper connection string to the database, but the service is conforming to the CA grid interface. It's at least read our DLL. It would have barfed by now if we couldn't get that. So our big test here is to rerun the CQL client. We're going to cut and paste the service endpoint. Again, this is local development machine, but this could be Azure. And so we're going to take our existing client. We're going to type in the URL. We're going to hit the load model, cross our fingers. And so it went out and asked the service for the model. You'll see what came back is actually the same information as we had before hitting the other live model, the live services. And so here we can type in our same query, participant collection summary, add criteria. We're going to do the provides. Now I remember the original what I was trying to do. This is the prostate prostate gland query. And so we're going to update the query, and now we're going to submit it, and hopefully if we get a nice we get a nice table. So this is, what we've done is we've started from scratch, really. We've configured a service, we've stood it up, we've run our legacy client against it. Essentially, this is on CA Grid now, and it took us about 22 minutes or so to do this. Okay? Hold on, last slide. Right, so I hope it didn't go too fast. Good news about that was it was actually truly running code, no vaporware. We've used the model as a layer between the service code and the unmodified database. We haven't really exploited the really interesting capabilities of model-based development, but that model was particularly useful in what we were trying to do. This is something that the University of Colorado is trying to stand up today, as a matter of fact. Note, we could also start from scratch and do a whole model-based development and creation of a whole new database. That's really some capabilities that I didn't touch in here. It was a combination of new support in Visual Studio 2010, really nice support for modeling, and we had to use some code that we wrote to tap into that. I mentioned this briefly that clearly this checking that we're doing is not incredibly powerful at the moment, but we have the layering in there. And so we're going to work to create more sophisticated, what I call support for heterogeneity. In other words, what can we really do in that layer such that we can retain our old database and still conform to CA Grid principles. Hope I didn't lose you. Thank you.
Okay. We have time for one or two questions. Does anyone have any? Hold on. Gotta wait.
I've got a general one about Azure and cloud. Is what for this type of service? What's the business model for paying for the cloud services? Either whether Azure or whoever.
That is an awesome question. By the way.
So it costs money to ship data in and out of the cloud, whether you're using Azure, or Amazon, Google. So we're doing a lot of work. We're doing the same thing. But have the scientists actually thought about?
Right. So I did a really poor presentation with regard to Azure, and I apologize for that. I did not touch on about what Kenji is asking, which is an excellent question. Azure in general, the nicest way Azure could fit into CA Grid would be large-scale data processing. Clearly, they're not there. They're not ready for that, frankly. What we're trying to do is just sort of say Azure, 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 and get in the game that way. That said, there is actually a business model, in particular, the NCI organization. We did this about 6 months ago, where they said that they were trying to stand something up, and their sysadmins had to go through blah, blah, firewall this, port that, blah, get back to us in 8 months, right? And so a business model is clearly, if this in fact is there's no security constraints on the data itself, which there is not on this, then yeah, put it out there in Azure, and you have your information, and you don't have to deal with your local sysadmins. This is actually useful for the Colorado people right now. So the biggest case for Azure right now for the Colorado people is to get away from their stupid sysadmins. Not the most exciting reason, but it's a valid reason for them. And we try to hook them, and we'll get them interested in large-scale stuff. We hope.
So I recently learned about Entity Framework. Can you contrast this with Entity Framework?
That's an awesome question, too. Short answer is no. The longer answer is it's difficult because Microsoft just changed direction on the Entity Framework. Did you know about this thing called Oslo, which they recently killed?
No, I guess I don't.
Because Oslo was kind of competing with Entity Framework. They killed off Oslo, and they're trying to put some other stuff into Entity Framework. How Entity Framework fits into this is an awesome unknown question. However, a more trivial yet valid answer is this required UML. This required, here's a model, you conform to it. The model definition is in UML. Entity Framework does not deal with UML. Visual Studio does. Entity Framework, if you just have a database, you can generate a model very nicely from Entity Framework, and that's really cool, interesting technology. We needed UML in this particular. Again, where it gets fuzzier is where Microsoft. I'm in the University of Virginia. I don't know what Microsoft's going to do. To me, they should converge, but it's not sure. It's not clear when and if they will. So how they actually come together, when they come together, which one I go for which capabilities, I don't know.
Okay, so we're going to get ready for our next speaker, who is Paul Anderson from Wright State University. He will be talking about cloud-based MapReduce architecture for nuclear magnetic resonance-based metabolics.
Oh, waiting for it to go ahead.
Yeah.
Okay. I'm actually, I used to be at Wright State University, but now I'm actually at the Air Force Research Laboratory. So what we're going to be talking about today is some of the things that we've been doing in collaboration with Wright State University, also with the University of Cincinnati and some other institutions around the area, to develop services and tools for the toxicologists, for the molecular biologists, for all the people that work over at the Air Force Research Laboratory to study the things of interest to them. So first, what metabolomics is, which is what we're going to be talking primarily about today, is the global study, the global quantitative study of your metabolites. Your metabolites are your small molecules in your body. And the metabolism is basically the set of chemical reactions that maintain life. So what we're talking about here is measuring globally, possibly in, say, your urine or your blood or tissue, all the different metabolites, these small molecules that are responsible for keeping you going. And as far as scale in relationship to the other omics categories, it's relatively the new kid on the block. Genomics and proteomics are a little bit more well-defined, in my opinion, and a little bit more mature. But there can be some argument about whether or not where we should be spending the majority amount of our time, and it really has to do with the way genes are expressed, the way that genes are then translated into proteins, and then those proteins, which can kind of be viewed as your molecular machinery, how they operate or work on the metabolites. And if you are studying that lowest level there, you can really get an accurate picture about what's going on what's going on in your body, because things are expressed but not necessarily translated. Proteins are floating around your body that are maybe not doing anything with metabolites. But if you're studying that lowest level, there can be some argument among biologists about where we should be spending our time. Now, there really are two main techniques, experimental techniques, that are commonly used, although when I mention mass spectrometry, there are really a lot of different mass spectrometry machines. But in general, there's two ways that people go about metabolomics. They either use nuclear magnetic resonance or mass spectrometry. They're really complementary techniques. They each have their own advantages and disadvantages. Briefly, I'll just mention some broad ones for you guys. The sensitivity is much, much greater with mass spectrometry. But again, it depends on which type of mass spectrometry you're doing here. But in general, NMR is non-destructive. So your samples are, you can run your samples as many times as you want. It requires very little sample prep, but it's not as sensitive. It's very comparable. Inter-lab comparability is very high. I can send my sample over to Europe, and I'll get the same result that I get over here. With mass spectrometry, it's a little bit grayer there. There's calibration that has to go on, the different machines. So really, there's, in my opinion, they're both valid techniques. Whenever I go to give a talk to more of a biologically based audience, I'll get arguments and people standing up saying one technique is, NMR is dead, or whatever. Okay, we're going to bypass that and just suffice it to say we're going to be using nuclear magnetic resonance to measure those small molecules in your body. And some of the things that the military are interested in, I'll give you a couple examples. We do. One of the studies that I was working on actually this morning when I woke up, because I'm still on Eastern time, so I wake up in the morning and, wake up at 6 a.m. and it feels like I've slept in. But we're studying the effects of mustard gas on, obviously, for our soldiers, but we're studying it on animals. And so we're trying to develop therapeutics for mustard gas exposure. Other things that we're doing, we're studying neurotoxins. So we could be studying, say, like sarin gas or something like that. We study a variety of them. And to give you kind of a picture about what we're doing here, I have kind of a very crude drawing of two neurons over here. And what these neurotoxins, these organophosphates, essentially do is they're going to go in and disrupt the signaling here, the synaptic transmission between two neurons. And the way that they do that is actually by inhibiting or actually chemically modifying a enzyme called acetylcholinesterase. And basically, roughly speaking, acetylcholinesterase is a garbage collector. More specifically, it actually goes through and breaks up acetylcholine so that it can be recovered. But what the neurotoxin's doing is it's interfering with this enzyme so that it can't do its job. And what happens is you get this buildup of acetylcholine here, which essentially means that these two neurons are consistently firing, which will lead to seizures and other horrible things that can happen. So that's understood, at least at a certain level. There's still research going on, and there's still more to figure out right at that level. But when we're talking about metabolomics, we're talking about trying to study the global effects that's going on. We'll do this experiment in animals, and then what we'll do is we'll stack the animals and create tissue samples from their brains and run them through the magnets or the mass spectrometry to get a global picture of what's going on. Because there's a lot of secondary effects that you still see and are not well understood at all. So typically in our experiments, we'll have control groups, maybe multiple control groups, multiple treatment groups. I have it simplified here with one of each, but we could have multiple doses, multiple treatment conditions that we're giving our animals, wide variety of groups. We could be studying those across time, and what we're going to be generating as output are these spectra here that we then have to come in and analyze. And I have kind of this is actually a high dose of, I think, a kidney toxin that produces this result. And we can see that we get a large difference in some of these signals here, which, again, citrate and alpha-ketoglutarate are small molecules we see some radical change in, say, from control to treatment. And so in general, that's what the biologists want to know. They want to figure that out. So we're going to be talking about how to develop tools to help them accomplish that. But in order to talk about those tools and what they do, I have to give you just maybe a little bit more about the data. So for NMR specifically, if we were to, say, measure one of our favorite small molecules, alcohol, in the magnet, we would actually get, if we're running proton NMR, there's proton NMR, carbon NMR, phosphorus NMR, there's different types of NMR. But if we're running proton NMR, we're going to actually get a signal for this molecule from each group of hydrogens here. And so the only point I'm trying to make here is that the data has some built-in redundancy, which is good and bad. There are redundant signals that we can try to measure to get a more accurate quantification. But what happens is when you go to an actual sample from urine or blood or something like that, you have a lot of signals in there. Because each individual metabolite could give you 6, 10, 20 signals depending on the metabolite. And so if you start zooming in on this, you just kind of see how complicated it is. And this is kind of ignoring, this is just one sample over here. This is ignoring any signal processing issues or variability between samples, which is what I'm talking about over here. We only have, I think, probably 10 signals here, but there could be variability in shift, and that shift could be arising from different sources. There could be variability that's introduced, baseline distortions. There could be a lot of issues going on here that we'll have to deal with before the biologist can do what we want to do on that previous slide and say, okay, citrate changed. Okay, there's a lot that goes on before we can get to that point. So the way that they tackle it, at least at the majority of labs and people and what we're trying to contribute to, is they either do maybe a combination, usually probably a combination of what I have up here at the top, where they're using some proprietary software, which is actually quite good. We actually use Economics, which is a software suite, in our labs, and it's very nice, but it's also very frustrating as a computer scientist because I can't extend it, I can't really interoperate with it, I can just use it as a black box. It can process some things, I can save them to files, and maybe I can take off where it left off, but in general it's very frustrating for me. The flip side of that is we can do everything kind of in-house with our own little scripts and our own little tools that the biologists have accumulated. They're good at Excel, they're possibly good at MATLAB or something like that, so they're good at a few things that they like to use over and over and over again. But this will inevitably fail because the tools just really aren't domain-specific enough for them. Like Excel, we're dealing with the data I was working on this morning was mass spectrometry output that a collaborator sent me in Excel, and it's just this huge table of all these proteins with all these replicates, and it's just he wants me to find the patterns in there for him. I mean, that's basically just here. So not really set up to do what he needs to do. We're going to come in and try to introduce this cloud-based software architecture where the scientist can basically be anywhere as long as he has an internet connection with his laptop, access our services, use a scientific workflow engine such as Taverna here to interact with the services and process his data. And our architecture is set up like this. We have our clients using Taverna at the top. Taverna is actually the data management and data flow management here for us. It's going to be calling our web services. Now, we've wrapped them using Ruby on Rails, but that isn't really what the talk is focused about. You could use anything that you want there to wrap your services. We're going to basically talk about how we've implemented those services using this Apache Hadoop Java framework here, which is I think most people are familiar with it, or at least have heard about it. Very widely used MapReduce architecture for computationally intensive and large parallelizable data sets. It includes the Hadoop file system, Hadoop distributed file system, which is very nice for our application dealing with these large data sets. So the reason why we want to do this is because a lot of our pattern recognition or signal processing techniques that we're running here are very easily and intrinsically parallelizable. So we can use the MapReduce paradigm to really give us a huge performance increase without actually spending time learning, MPI, or if we're using MATLAB, figuring out how to set up a MATLAB cluster and so on. I can just use what's already built in there for me. We can either, at a very rudimentary level, I can either say, I just want to, if I have hundreds of spectra, maybe I just want to operate on each one of those independently. But we'll see some other applications where we can actually divide up the spectra, process those in regions, combine them using the reduce phase, and so on. So first, a really simple one that even though it's a simple task and simple algorithm, we see a large performance increase when we start getting multiple users on our systems, a user that has a lot of spectra, and so on, is just a very simple normalization techniques where all we're going to be doing is trying to correct for, say, urine concentration differences. Specifically here, I just kind of have scale. These are actually the same image here, just kind of scaled up and down. And we're just going to make sure that each of these spectra sum to a constant value. So thus the name, constant sum normalization or so on. So it's a simple technique, but if you're doing it on hundreds of spectra, it'd be nice if you could just farm that out and just say, do it in parallel, okay? And because it's a relatively simple technique, we can actually use something that Apache has created called Pig Latin. Pig Latin is a language that sits on top of Hadoop that you can write without necessarily worrying so much about the underlying MapReduce implementation. We can do our sum normalization in just these 8 lines of code-ish or something like that, just by doing some nice Pig Latin script where we're grouping things a certain way, summing them. They're going to translate that automatically for us into the Java code that we would need to run on Hadoop. And it does it all in the background for us. So we can do this for Pig Latin for a lot of our things that really are that simple. I mean, once we get to some of the other applications we'll talk about in a bit, Pig Latin's not going to be sufficient for our needs. But it is widely used at Yahoo. 40% of all the Hadoop jobs that are there actually run with Pig. So it can do a lot. It's just sometimes it's insufficient for what you need. 5 minutes, really? I've been talking way too much. Another application is baseline correction. And this one, we're basically just trying to correct for distortions that we see in our samples here. The algorithm is a little bit too much for implementing, say, using Pig. So what we're actually going to do is implement our own solution. And we're actually going to take advantage of another Hadoop feature, which is called Hadoop streaming. Because I'm traditionally on bioinformatics. eScience is a relatively new thing for me. It's been a year or so we've been working on this. And so I had this algorithm already written in MATLAB. MATLAB's great at doing matrices and so on. Well, I just want to put it up there on our cloud, on Hadoop. Well, all I need to do is just actually compile my MATLAB code into an executable, and then I can use Hadoop streaming to run the job for me. I don't actually have to know it, you translate it to Java. All I need is my application to read and write from standard in and standard out. Another one that we have implemented is a deconvolution algorithm. So in the end, we're going to want to quantify those individual signals to say, here's citrate, here's alpha-ketoglutarate. What are the actual concentration values of those? So this is even a more data-intensive algorithm that, again, we originally had written in MATLAB, but we can actually, it can become computationally feasible by using the cloud. Something that could take a day or two to run can now be run on the cloud in just a fraction of the time. And here I just have some details about the algorithm that are, not necessarily important for this talk here. And again, we do this one using our streaming interface in Hadoop. I got my code in MATLAB. Again, I'm traditionally a very good MATLAB programmer, so I can prototype something very quickly. It's research-oriented, so I want to get something out there for the researchers very, very quickly. All I need to do is compile it down, decide what my mapper phase is going to be, decide what my reducer is going to do, and I have my application. And we see, huge performance gains. I mean, this is just with the sum normalization where if you're dealing with any kind of large data set, you quickly overcome the performance of, say, a single machine or even a multi-core, multi-processor machine. And so in summary, what I've basically described is just some of the things that we're trying to implement for our scientists to use over at the Air Force Research Laboratory so that they can basically do their science a little bit faster and a little bit more efficient, a little bit better. We use Rails on top of our Hadoop implementation to expose these to Taverna, which the scientists can then interact with in a workflow environment. And some of the future things that we want to do, so this is very Air Force research focused right now, and they're not necessarily concerned that we make this available to the general public and so on, but that's where we eventually want to go, put it on some publicly available clouds. I mean, obviously our code you can download and things like that when we publish, but as far as getting this out to the public, we still need to work on that a bit. And obviously just more and more services. I mean, a lot of techniques that have been developed and a lot of techniques that are out there that we'd like to plug in so our scientists can use them. And obviously, I just want to acknowledge my collaborators at the Air Force Research Laboratory, at the Noesis Center, at Wright State University, and Microsoft Research for really, this is my first time here. It's been a great workshop, very educational. That's it. Thank you.
So, do we have any questions? Any?
I gotta ask about Dryad. Did you try
I'm sorry?
Dryad? Have you tried Dryad?
Not familiar with Dryad. What is Dryad? Again, I'm a computational scientist, not necessarily an eScientist. It's a new thing for me. But what is Dryad?
Do you want to answer that one?
It's the Microsoft equivalent of Hadoop.
Oh, is it? Oh, okay. No, we haven't tried. I think the reason why we ended up using Hadoop is one of our collaborators, Dr. Kiki Chan. He actually was one of the developers with Hadoop at Yahoo before he came to the university. So I don't know. Is Dryad better? I don't know.
I assumed that it would work well on your application. You're welcome to try it out if you wish. We can get you access to Dryad instances.
Well, that'd be great. I'd love to try it.
Yeah, they use Dryad in the Worldwide Telescope.
Oh, okay.
So it is ideal for a computational large data set. So do we have any other questions?
Just in general, this is not dissimilar to what we go through at Illumina. Did you see improved accuracy, even though you've cleaned up the workflow? Does it help improve the accuracy because everything's consistent?
Well, we see it at a lot of different levels. One thing that we saw when we were first creating these systems for our biologists is just samples are mislabeled, they're misclassified, they don't have all the samples, and because we're dealing with large studies here with lots of different animals, and it's like, well, did that animal die? Did it become sepsis? Should we throw out that entire animal? But using these services instead of these Excel spreadsheets or these proprietary software really allows the scientists to take a look at it and find those errors, and we've resolved them and gone forward. So that's been great. As far as the accuracy of the algorithms are concerned, I mean, running them on the cloud doesn't necessarily make those any more. It just makes them faster and available to the scientists. So maybe it makes their work more accurate because they can use the tool that they couldn't before, which is maybe a better tool.
I have one question. What were some of the implementation challenges? You talked about how easy it was, but what were some of the challenges you had?
Okay, so there was, MATLAB in particular was a frustrating thing to work with just at the beginning because I don't know if many people have used MATLAB, but again, it's supposed to be this development environment for scientists, not necessarily a standard programming language. So Hadoop really wanted to be able to read and write from standard in and standard out. Well, writing to standard out was not a problem. When you compile something in MATLAB, you can easily write to standard out. But reading from standard in was another thing because MATLAB, they already capture standard in, so they don't want to give you access to standard in. So we actually had to do a little bit of I don't want to call it kludge or anything like that, but just something that shouldn't be necessary but was necessary because of the way MATLAB does it. We had to actually write a little C wrapper before we called our MATLAB code. But the only thing that does is just grab standard in and pass it to the MATLAB function so that it can actually run with Hadoop. That was the biggest, frustrating implementation issue.
That concludes this session. Thank you for joining us. And we'll be starting session 4C.